Good morning, good afternoon, good evening, everyone. Welcome to this Inside Scientific webinar entitled We Are More Than What We Eat, Dietary Interventions Depend on Sex and Genetic Background. My name is Darius Sulam from the events team at scientist.com, and I'm pleased to be your host today. This webinar has been sponsored by Columbus Instruments, so a big thanks to them for helping to produce the webinar and making it possible for us to share science together. We're fortunate to have Dr. Dudley Lamming, who will discuss the importance of sex and genetic background in response to diet. Hi, Dudley. How's it going?
Great. Thanks for having me.
Absolutely. We're absolutely thrilled that you're here to give this presentation. And I'm going to just pass it off to you whenever you're ready. Take it away.
Thank you. What should I eat? It's a question that we think about every day. Noted food author Michael Pollan said that humans have an omnivore's dilemma, the fact that we can choose from a wide variety of different types of food. And of course, these may vary in their effects on us in terms of our health. When asked what we should be eating, he said that we should eat food, not too much, and mostly plants. And certainly the part about not too much is something that I think all of us are aware of. There's an epidemic of obesity and diabetes going on around the world. And conversely, over a century ago, it was shown that eating less food can be beneficial for the health of animals. Here on the left is data from mice, looking at mice that either were fed an ad libitum diet, they're able to eat whenever they wanted to, they had free access to food, and then animals that were restricted by various amounts. You could see that there was an enormous increase in longevity in these animals when their calories were restricted by about 40%. And more recently, work from Ricky Coleman and Roz Anderson showed that similar effects can be seen in non-human primates. Importantly, calorie restriction doesn't just extend lifespan, it also extends health span. And so it allows animals to live healthier lives where they have less cardiovascular disease, cancer, diabetes, and in models where this is a problem, things like Alzheimer's disease as well. Unfortunately, many people, most people probably, are not going to be able to adhere to a calorie-restricted diet because there's food constantly surrounding all of us, and people who are on calorie restriction definitely are hungry and suffer from other side effects related to the fact that they are eating so much less food than they normally would be. So we've been thinking more about dietary composition. What is the food actually composed of? And a diet has three major macronutrient categories: protein, fat, and carbohydrate. And for many years, there's been this idea that dietary fat, in particular different types of fat, might be bad for us. More recently, there's been a lot of attention in the press and by diet gurus saying that we should be eating less carbohydrates, particularly really refined carbohydrates, that this is contributing to the epidemic of diabetes and obesity. But in all of this, dietary protein has been viewed as generally neutral or positive. First of all, dietary protein promotes satiety, so it helps us feel full. And so if we eat more protein, that might make us eat fewer calories. And simply, even if we don't wind up eating less, if we ate more dietary protein, this would crowd out fat and carbohydrates. And so as a result of this, dietary protein intake has been heavily promoted both in various types of fat diets as well as by governments and medical groups, saying that we should eat large quantities of protein, or at least certainly not cut back. But this doesn't seem to be the case from an epidemiological standpoint. Low protein diets are actually associated with decreased rates of cancer and diabetes, as well as in people under the age of 65, lower mortality. And a very large prospective cohort study in Europe, following about 40,000 people for a decade, found that diabetes risk actually doubles with increased consumption of protein. And a number of other studies similarly have suggested that high protein diets might be associated with poor health outcomes like increased cardiovascular mortality. To try and understand this a little bit better and to do this as rigorously as possible, we collaborated with Luigi Fontana whose laboratory at the time was at Washington University in St. Louis. And the people there were placed into one of two groups, either a control group that ate their normal diet at home and wrote down what they ate, and a protein restriction group where the calories that came from protein were decreased by about half, from about 17% in the control group to about 7 to 9% in the protein restricted group. And they were followed for about 6 weeks. And the thing that was really interesting from a metabolic perspective was that people in the protein restricted group lost weight. So over the course of 6 weeks, they lost about 2.5 kilograms, so a little over a pound a week. And a slight majority of that was from fat mass. And so this is not just lean mass or water weight loss. They're literally having less adipose tissue as assessed by DEXA. And other health improvements were observed as well. For instance, a decline in fasting blood glucose levels. Now, whenever we think about a dietary intervention in humans, or as I'll be talking about next, mice, we have to think about the fact that maybe the diet has palatability issues, for instance. And so these diets are quite different. And so we wanted to see if these people were losing weight due to eating less. And interestingly, this turned out not to be the case. Now, you'll recall that earlier I said that protein is satiating. It helps us feel full. And we observe that as well here. So the people who are on a low protein diet, who are eating less protein, could eat whatever portion they desired in the hospital cafeteria. And their calorie consumption actually went up by about 10% versus baseline. So these people were eating more, but they gained less weight. Now, for most of the rest of this talk, I'm going to be focused on mice. And why should we study diets in mice? So there are a lot of advantages to this. First of all, mice eat exactly what we feed them. When we are doing human studies, for the most part, any long-term study in particular, people are exposed to food in the environment and constant temptation. So there's always the possibility that they're going to eat something that they're not supposed to by the study design. We can measure exactly how much they eat. This is always a big problem in diet studies. People say that they, ate a certain amount of food, but did they weigh it? Did they measure it? How do we know exactly how much of it they ate? It's a big burden for people to track that, and a big problem for researchers to follow up. We can control diet composition precisely. And so this is something that I think is very important, because when we're looking at dietary macronutrients in the context of human food, a lot of things change. So we're talking, for in the case of protein restriction diets, for people who are eating sort of a typical American diet, burgers and frankfurters or whatever, and switching to something that's very healthy, right? Sort of fish and legumes and greens. And so in a mouse context, we can eliminate those confounders. And we could do a lot more tests. Mice we could study for a longer portion of their life, and we could do tests that are more invasive. And so to summarize a lot of studies from our laboratory that I've talked about previously and that have been published over the last few years, what we found is that mice and humans tend to respond very similarly to protein restriction. They have lower weight and lower adiposity overall, and significant improvements in glucose tolerance. And so those have been published in a number of studies, and most recently a paper in Nutrients showed that protein restriction seems to benefit the health of humans with type 2 diabetes as well. And so overall, we found that this is very beneficial. Now, one issue with mice that is not, which is sort of a weakness in studies, is that these studies are often done in only one genetic background, most typically C57 black 6 mice. And over the last few years, it's become clear that genetic background seems to play a very important role in the response to dietary protein and other nutrients. And so each in an ideal case scenario, right, humans are all genetically unique. And so ideally, we'd be able to study these types of interventions in genetically unique mice as well, or at least panels of mice that allow us some reproducibility. So our question that I'm going to tell you about first is, how does genetic background contribute to response to dietary protein? So in order to study this, we used 3 different mouse strains. The first 2 strains we used are C57 black 6J mice and DBA2J mice. And these are 2 different inbred strains, and we used both male and female animals. The third strain that we used was Het3 mice. So Het3 mice are genetically heterogeneous. Every animal is unique. But they're derived from 4 different founder strains in a series of defined crosses. And so the F2 population, even though every animal is genetically unique, the population is reproducible. And we used 3 different diets. So our control diet was 21% of calories coming from protein. Our low protein diet was 7% of calories coming from protein. And then we had a medium protein diet that was right in the middle, 14%. And the diets here are all isocaloric. They have identical levels of fat. And so the calories were matched by increasing carbohydrates. So our low protein diet here is a low protein, high carbohydrate diet. So one of the first things that we did over the course of about 3 months was track how these diets affected weight and body composition. And the thing that's really interesting here is that there's quite a bit of variation both between strains and sexes. So here you can see that low protein diets cause less or associated with less weight gain in these young growing animals. These studies all started about 10 weeks of age. And you can see that these mice gained significantly less weight than mice that were on the control or medium protein diet. But we didn't observe those differences in females. And conversely, when we look at DBA mice, we did observe affected females, but not in males. And then our genetically heterogeneous population showed significant differences in males, but no differences at all in females. And this is reflected overall in changes in body composition as well. There's differences in between sexes and strains in terms of gain or loss of fat mass, gain or loss of lean mass, and changes in adiposity. We also see this type of variation with respect to other phenotypes. So these are glucose tolerance tests. The mice are fasted overnight and then administered glucose at time 0. And what we see is we're tracking what happens to blood glucose levels over time in these mice. You can see that C57 males that had reduced weight gain do indeed have improved glucose tolerance. But we actually see changes in females as well, even though the females didn't have that effect on weight gain. And then Het3 males have quite a profound response to dietary protein. Even the medium protein diet here improves glucose tolerance. But again, DBAs do not, and then females, Het3 mice don't either. So there's quite a bit of variation. Now, I'm not going to take you through every one of the tests that we did. In this particular study, we did over 30 different assessments of physiology, both body composition, weight, glucose tolerance. And we used metabolic chambers as well to measure food intake, energy expenditure, and track activity. And what we found was that some effects were very strongly either positively or negatively associated with dietary protein intake across all 3 strains. And these include things like effects overall on fasting blood glucose levels and lean mass. Negative things that were negatively correlated included things like energy expenditure during the light period and the dark period, measured using our metabolic chambers. And then here in the middle were some of the most interesting things where we're looking at things that are very heterogeneous between the animals. And these included a number of things that we'll talk about, particularly a hormone called FGF21. Another way of looking at this might be to do a principal component analysis on the various phenotypes. And what you can see here is that we do indeed have quite a bit of variation between strains. So C57 and DBAs are sort of the most distinct, with Het3s somewhat in the middle. And there's quite a bit of distinctiveness between males and females as well, although there certainly is some areas of overlap. Another way of looking at this is pathway analysis. So this is pathway analysis that we did on transcriptional profiling from the liver of C57 and DBA mice on either control, low protein, or medium protein diets. And of course, there's a lot of pathways here, and the text is very small. But the thing I want to point out to you is that when we look at mice that are looking at the effect of a low protein diet, here in B6, we have tons of pathways that are changing significantly in the down-regulated direction or up-regulated direction. And there's differences between the males here on the left and the females here on the right. If we look at DBA, however, we see that the number of pathways that change is much smaller. And that sort of correlates with what we see in our glucose tolerance tests, as well as our effects on weight. A lot of the phenotypes are distinct between B6 and DBA. And then if we look at our medium protein diet here on the right, what we see is that there's actually almost no effect at all of any of these changes. And overall, this reflects a lot of the phenotypes as well. So our medium protein diet actually has very few molecular or phenotypic effects. So we wanted to know if we could integrate both our transcriptional profiling data as well as our phenotype data to gain new insights into what dietary protein is doing. And so we actually combine not only transcriptional data and phenotype data, but also liver metabolomic data and lipidomic data between Black 6 and DBA mice on either a control diet or a low protein diet. And looking at the things that changed significantly in each of these, we did a correlation analysis and then clustering. And this gave us 6 distinct clusters, each associated with certain phenotypes, as well as pathways that change both at the transcriptional level here in blue, metabolomic level in green, or the lipidomic level in red. Now, I brought your attention to a hormone FGF21 before, and FGF21 has quite a bit of variation between strains and sexes. We'll look at that a little bit more. But one of the things that sort of jumped out at this was that FGF21 is associated with just one very small cluster here. And the reason that's so interesting is that FGF21 has been reported as a key mediator of the effects of dietary protein restriction. So FGF21 in Black 6 male mice was shown to be an endocrine signal of protein restriction. It's induced by protein restriction in those mice. And also, if we use the investigators here, Chris Morrison's lab and some others, knock out FGF21, the effects of a low protein diet on mouse metabolism is ablated. And this has been worked out in quite some detail, looking at how FGF21 from the liver actually signals into the central nervous system of Black 6 male mice. And what we found is indeed that Black 6 male mice have a profound response to dietary protein with respect to FGF21. You could see that the levels of FGF21 about triple or more on a low protein diet compared to a control diet. But interestingly enough, this seems to be a strain and almost sex-specific effect. In DBA mice of males and females, there's no induction of FGF21 on a low protein diet. The same thing is true in Het3 mice, even the Het3 males that actually have quite a profound effect of dietary protein on glucose metabolism and body weight. C57 females do seem to have a trend here, if it wasn't statistically significant, but again, you could see that it doesn't reach the same scale as we see in the Black 6 males. So in order to dig into this a little bit further, we took mice that were conditional for FGF21 and knocked out FGF21 in all their tissues. And we then fed these mice or their wild-type litter mice to either a control diet or a low protein diet. And we observed right away there are differences in response to body weight. So as we expected based on our Black 6 data, what we see is that low protein diets are associated with essentially no change in body weight in these young growing mice, whereas in wild-type animals, either the control diet or both the control and the low protein diets are associated with body weight gain in FGF21 knockout males. In females, however, it's quite a different response. Here in females, FGF21 status doesn't seem to actually change at all what happens to body weight gain. The mice overall that are knockouts are a little bit lighter to start with, but they gain weight at the same rate as wild-type animals. Now, to try and understand the basis for this, we placed these mice in metabolic chambers from Columbus Instruments. And what you could see here is we were measuring food consumption and energy expenditure in males over the course of a day, 24-hour period. And what we see is that in wild-type mice, there's quite an increase in food consumption on a low-protein diet. This is something that we expected to see based on many previous studies of ours, but also, again, it fits with the satiating effect of dietary protein. And in the knockout mice, that was completely blocked. In terms of energy expenditure, we see something very similar. Energy expenditure is increased in mice fed a low-protein diet. That explains why they gain less weight. This seems to be associated with browning and beiging of white adipose tissue, and the details of this have been worked out quite nicely by Crystal Hill. But in the knockout mice, this increase in FGF21 is disrupted. So this all fits with the paradigm that FGF21 is a critical mediator of the effects of dietary protein. But in females, it turns out that that's quite different. So in females, indeed, we do see an increase in food consumption in wild-type mice when fed a low-protein diet. But in knockout mice actually have an increase in food consumption all on their own that's suppressed by a low-protein diet. So the opposite of what happens in males. And when we looked at energy expenditure, there is no effect of dietary protein in either the wild-type mice or the knockout mice on energy expenditure. And so overall, this seems to be quite distinct in terms of what we're looking at. So potentially, FGF21 may only be important for response to PR in black 6 male mice. And this is something that we're investigating further, looking at a variety of different strains, and I'll tell you a little bit about that later. But we really wanted to know as well, what is it about a low-protein diet or protein-restricted diet that promotes metabolic health? And so proteins are made up of amino acids. Typically, we think about the 20 common amino acids that make up our diet. And we've focused on the essential amino acids. There are 9 essential amino acids for both humans and mice. They're the same set. And a lot of our work is focused on the branched amino acids, leucine, isoleucine, and valine. Now, BCAAs have been known to be interesting from a metabolic perspective for some time. Back in the 1960s, it was shown that they're elevated in humans who were obese or insulin resistant. And subsequent to that, it's been shown that BCAAs are associated with obesity and insulin resistance in multiple rat and mouse models of metabolic syndrome. And in blood seem to be very well correlated with glycated hemoglobin levels as well. And interestingly enough, when animals tend to lose weight, their branched amino acid levels tend to go down. And this is true for humans as well. So in our protein-restricted human study, we found that blood levels of the branched amino acids declined by about 10% or so. And interestingly, this was specific for the branched amino acids. So even though they're eating less protein overall, other amino acids in the blood, such as methionine, which has been associated with some of the benefits of a vegan diet, actually didn't change at all, even though the branched amino acid levels declined. So we found in some of our published work that low branched amino acid diets could recapitulate some of the benefits of a protein-restricted diet. And more recently, we've dived into trying to understand which of the branched amino acids really mediate this effect, taking mice and placing them on a series of diets where everything is as controlled as possible. So the diets are isocaloric, they have the same amount of fat, they have the same amount of carbohydrate, they have the same amount of essentially nitrogen, so the diets are isonitrogenous. We've restricted each of the diets here by 67% when in terms of leucine, isoleucine, or valine, or all 3 branched amino acids, and then increased the amount of a non-essential amino acids proportionally, so that the diets are isonitrogenous as well as isocaloric. So we're trying to control things very well here. And interestingly, in black 6 male mice, we see that there's a very strong response to dietary isoleucine restriction. So restricting isoleucine recapitulates or more all of the benefits of protein restriction, significantly improving glucose tolerance. Here's the red area under the curve for the low isoleucine fed animals compared to the control diet fed animals, which is much higher. And interestingly enough, leucine, which is the most abundant of the branched amino acids, had almost no impact. And subsequent studies further restricting the amount of dietary leucine confirmed that leucine is not really a key mediator of these metabolic phenotypes. To a lesser degree, there is some effect of valine, particularly in diet-induced obese mice. Now, trying to figure out the metabolic basis for this, we took advantage of our collaboration with Joseph Bauer's lab at UPenn to use hyperinsulinemic euglycemic clamps to measure hepatic glucose output in the basal condition as well as in the presence of high levels of insulin. And what we found was that in the fasted state, control-fed mice and low isoleucine-fed mice, their livers produce similar levels of glucose. But in the presence of insulin, control-fed animals have a modest suppression of hepatic gluconeogenesis. Well, there's much greater suppression of hepatic gluconeogenesis in the low isoleucine-fed animals. So one of the major differences here is that low isoleucine-fed animals have significantly better hepatic insulin sensitivity than control-fed mice. Isoleucine seems to correlate with a lot of the other phenotypes that were associated here as well. So mice fed a low isoleucine diet tend to gain much less weight as well as fat mass compared to mice in the other diet groups here, particularly the low leucine group or the control amino acid diet group. And again, there seems to be not too much of an effect of dietary valine, but a little bit of an effect. And so looking across different metabolic phenotypes that we examine, what we see is that low isoleucine and to a lesser degree low valine really recapitulate the benefits of a low BCAA or protein-restricted diet, whereas leucine's effects are quite distinct. Now, today's theme is talking about genetic variation, and so we've begun to look at isoleucine restriction in the context of a variety of different strains and sexes. Interestingly, unlike protein restriction, which seems to be very strongly varied between sexes and strains, isoleucine is a bit more potent. And so isoleucine restriction improves glucose tolerance here in black 6 males as well as in females, fairly well in DBA males as well in the context of a Western diet. These are all Western diet fed animals. And there's even a trend in the similar direction, although it didn't reach statistical significance in the DBA females. And so this seems to potentially be a little bit more universal effect than protein restriction was. So we're really interested in trying to understand the mechanisms behind these effects. One of the mechanisms seems to be increased energy expenditure. And we see browning and beiging of white adipose tissue in low isoleucine fed animals. When we looked at the gene expression in the tissue here, we saw increased levels of not just UCP1, which is a critical mediator and marker of beige adipose tissue, but also other thermogenic genes. And then using our metabolic chambers, we were able to quantify the effect of these various diets on energy expenditure. What we found was that both low isoleucine and low valine fed mice had increased energy expenditure, correlating very well with the effects that we see on white adipose tissue beiging. Now, we took our FGF21 knockout mice and put them on either a control diet or a low isoleucine diet. And what we found for our B6 male mice is that there's this very substantial increase in wild type animals in terms of food consumption that's completely ablated when we knock out FGF21. And when we look at energy expenditure, the effect is not totally FGF21 dependent. Here we see a very fine difference in the dark phase, but about half of the effect on energy expenditure that we see is mediated by FGF21 and suppressed in the FGF21 knockout mice. So how do sex and genetic background contribute to the effects of a low isoleucine diet? This is a project that is unpublished data. We just put this up on a preprint server last week or 2 weeks ago. And we took our Het3 mice, or genetically heterogeneous animals, and tested what happens when we do this when we're young or when they're old. And what we see here in the young mice is that low isoleucine diets actually have a quite distinct effect from a low amino acid diet, where we're restricting essentially all of the dietary amino acids and essentially mimicking the effects of a low protein diet. What we found is that in our low isoleucine fed animals, there's a dramatic effect on body weight gain in the animals. And we don't see that to the same extent in the females, or in terms of a low AA diet either. There's certainly a little bit of a trend towards less body weight gain, but the low isoleucine diet is quite much more powerful. Overall, there seems to be quite a bit of variation between sexes and strains in their response to diet body composition. Here we're using an echo MRI machine to measure fat mass and lean mass. Fat mass is over here on the left, lean mass on the right. And what you can see is a low isoleucine diet certainly has pretty dramatic effects, suppressing fat mass gain over the course of about 3 months. And that's true in both males and females. A low AA diet seems to suppress body weight gain in males as well, particularly lean mass. And there seems to be sort of a trend towards that on fat mass as well. But this here is in the females. But just like our low protein diet fed animals and our low isoleucine fed black 6 males, we find that these animals are actually eating quite a bit more. And so that's true for both the low protein variant as well as the low isoleucine diet. So we fed to the Het3 animals. We also quantified isoleucine consumption, and you can see that isoleucine consumption is very similar between low AA and low ILE fed mice. So why do these animals eat less or weigh less? Well, just as in our previous study, we thought that if they are eating more and gaining less weight, then presumably there should be an increase in energy expenditure. And here we're plotting energy expenditure over the course of a 24-hour light-dark cycle. And you can see that in both males and females, both a low AA diet and a low ILE diet cause a significant increase in energy expenditure both during the night and during the day. And one of the things that's really interesting here to think about is that the low AA fed mice here didn't show nearly the same effect on body weight that the low ILE fed animals did, even though they have very similar effects in males on energy expenditure. And so that may be due to differences in the diets, potentially differences in activity or food consumption that are very fine. And so we're still sort of analyzing the data from our metabolic chambers to see if we can try and better understand why it is that these have very similar and dramatic increases in energy expenditure, but different effects on weight. We find that these diets also improve glucose tolerance. Interestingly, in the males, a low isoleucine diet improves glucose tolerance, while a low AA diet did not, but both diets did in the female animals. And so this seems to be quite interesting. And there's also a difference between Het3 animals and black 6 mice that we've looked at some length earlier. We started a lifespan study as well. And so my lab is really interested in aging. And so we took these mice at about 6 months of age. Typically, people would believe that's about 25 to 30-year-olds for a human being, and randomized them to either control low AA or low isoleucine diets. And we tracked what happened to their body weight as well as body composition over time. And as we expected, low ILE fed mice tended to weigh substantially less than control-fed animals. Low AA diets had some benefits for weight as well. And a lot of this was due to effects on fat mass. And so both low ILE and low AA diets promoted lower fat mass, particularly in females, for the low AA diet. There are also effects on lean mass, though, as well. And so definitely this is something to think about, right? I mean, we may not want to necessarily have interventions that cause significant amounts of lean mass loss, particularly in people who are older and, at risk of sarcopenia. So we have tracked this over time as well. We continue to see that low ILE diets are beneficial, even in quite aged animals. And improving not only glucose tolerance, but in males at least, insulin sensitivity. And this is something that we did not originally expect due to our previous data that we've looked at, but there were improvements in insulin tolerance as well. So looking across sexes and ages, we see that there are age and sex-dependent effects of a low isoleucine diet. Again, some things strongly correlate with dietary isoleucine intake. Some strongly correlate against dietary isoleucine intake, such as energy expenditure and RER during the dark phase. But definitely there are some things to think about, and some heterogeneity in particular in the middle here. Now, interestingly, we originally showed you data suggesting that low protein diets don't induce FGF21. And the same is true for a low isoleucine diet. And we did not see FGF21 induction in either young mice or old mice on either a low protein diet or a low AA diet. So the effects that we're seeing here seem to be completely independent of the effects of circulating FGF21. One thing that is really interesting, I think, is that a low isoleucine diet also reduces frailty. So when we first started working with these diets, branched amino acids and protein are typically thought of as something that's really associated with being strong and muscular. And there's concern that restricting these types of amino acids would make mice more frail. In fact, the opposite seems to be true. Control diet fed mice, particularly males, are frailer than low isoleucine diet fed animals that are fed in parallel. And this correlates very well with what we see with longevity. Control diet mice live substantially shorter than low isoleucine diet fed animals. And in particular in males, low isoleucine diet fed mice live much longer. So you can see an increase of 33% here in median lifespan, a very substantial increase in maximum lifespan as well. In females, we did see a statistically significant effect, but much smaller, about 7%. Now, people are always really interested in knowing whether this is true in humans as well. And we can't answer that question directly, but I can tell you that at least in people who live in Wisconsin, the amount of isoleucine in the diet correlates with body mass index. And so the thing the epidemiologists are really interested in is this beta equals 2.5, suggesting that as one's isoleucine intake increases from 4% of protein to 5% of protein, your BMI might go up by 2.5, which is about 5 to 10% of an individual's BMI, depending on what their BMI might be. So we think isoleucine, which is low in things like the flightless emu, but other things such as turkey, tend to be associated potentially with body mass index. And isoleucine may be associated with mortality in humans as well. So in a machine learning paper that was published a number of years ago looking at potential biomarkers for mortality in the blood, it was shown that isoleucine is actually associated with increased levels of mortality in people. And this is the only one of the 3 branched amino acids that is associated with mortality in that way. So in conclusion, sex and genetic background are critical regulators of the response to dietary protein, and probably isoleucine as well. And this is something that we're beginning to take advantage of to do genetic mapping studies. Here's some preliminary data showing that across a series of inbred recombinant lines, there's a very large heterogeneity in the response to dietary protein. Some mouse strains lose weight on dietary protein, but some gain weight on when we restrict dietary protein. And some mice have improved glucose tolerance. Many mice have improved glucose tolerance on a low protein diet, particularly in males. But a few get worse glucose tolerance. And which strain is where tends to be different between males and females as well. And so I think what this really highlights is the fact that for nutrition, mice are letting us to begin to take a look at how eventually, if we move these interventions to people, they're really going to need to be personalized. And something that I think is going to be really interesting is trying to take these animals and identify things like energy expenditure, activity levels, weight change, glucose tolerance effects in these different strains and do genetic mapping. And this is something that our lab is working on quite a bit. I'd like to thank everyone in my lab who has participated in these projects, Nicole Richardson and Charles Yu really led the initial studies on dietary protein and isoleucine. Kara Green has been doing the lifespan study for a number of years, and she and Mikhail Murphy have been investigating how dietary isoleucine protein affect health across a variety of strains and sexes. Also, our funders, the NIH and the VA hospital, as well as private foundations, are collaborators. Columbus Instruments for their sponsorship of this, and you for your attention. Thank you.
All right, Dudley, are you ready?
I'm ready.
All right. So the first question I have for you here is, protein diet tends to give the feeling of more energy. Did you notice any behavioral differences in terms of overall activity level? And would you expect routine exercise to significantly change these effects?
Yeah, that's an interesting question. I think overall, the general idea seems to be that satiety is directly sensed or regulated by dietary protein. And that didn't seem to correlate with activity levels very much. Now, you might expect that animals that are eating more and visiting the feeder more frequently might have additional activity as a result of that. And certainly that's one possibility. We have noticed some shifts in feeding time, and one thing that might be interesting to look at is whether or not they're spending more time feeding in addition to having that shift. So I think that would be a good idea to look at, and something that we could certainly do with our metabolic chambers. We do think that exercise will change some of these events, and we've sort of puzzled this out over a number of years because I think sort of the conventional treadmill tests might not be ideal for seeing these results, just because if you run animals frequently, just like if you run a person, frequently, they tend to lose weight. And so that's something that we will be looking into in greater depth perhaps for next year, because we started doing some resistance exercise training that might better model some of the effects in humans.
Okay, perfect. Thanks for that. Another question for you. Are the low and medium protein diets kept isocaloric to the control diet?
Yes, they are. So in our studies where we're using low, medium, and high protein diets, everything is isocaloric, fat content is identical, and so our low protein diet is actually a low protein, high carbohydrate diet, which, sort of in conventional wisdom would be, very unhealthy for you, but clearly, in this case, the animals are perfectly happy and even healthier on this low protein, high carbohydrate diet. In some of our studies where we manipulate just individual amino acids, we can also keep carbohydrates fixed and keep things isonitrogenous as well by boosting the level of non-essential amino acids a bit. And so that's sort of an advantage of just manipulating one amino acid at a time. But if we do manipulate all of the dietary protein or dietary amino acids at the same time, then it's necessary that we make that trade-off with carbohydrates.
Perfect. Thank you. How exactly was energy expenditure measured?
We used Columbus Instruments metabolic chamber system, the CLAM system. Some of our first studies were done with the traditional CLAM system, and now we're using the CLAM's HC home cage system, which I think has some major advantages in terms of not having to put the animals in such a stressful artificial environment. We could just keep them in their home cages and sort of swap out the lids and do indirect calorimetry by measuring oxygen and CO2 consumption and production.
Perfect. Thank you. Question here. You may have addressed this, but if you calorically restrict a mouse but supplement with isoleucine to normal levels, do the health benefits of CR disappear?
I'm going to take that in a slightly different direction. So if you protein restrict an animal and you add back isoleucine alone, you blunt or eliminate most of the metabolic benefits of a protein restricted diet. So from the context of protein restriction, that isoleucine is not only sufficient to give you these metabolic health benefits and to extend lifespan, but is also necessary for the metabolic benefits of a protein restricted diet. Calorie restriction is different though. So calorie restriction, we are calorie restriction maximally, we usually do about 40%. We've settled on 30 as something that is probably a little bit better and healthier for the animals in terms of a lot of metabolic adaptation. And so we assume that isoleucine levels there are going to be reduced by 30% as well. And that's probably not very much compared to the 67% restriction that we do in our protein-restricted diets. When we add back branched-chain amino acids to a calorie-restricted mouse, almost nothing happens. There's some very modest effects. And I would say that they are actually probably so small as to be biologically irrelevant. So we think that BCAAs and isoleucine contribute to the effects of protein restriction, but not necessarily to calorie restriction.
Perfect. Josephine says, thank you for this great presentation. She was wondering if you performed microbiome analysis and how it changes with diet.
That's a great question. So we did 16S rRNA profiling, and there are massive changes induced in the gut microbiome by either protein restriction or branched-chain amino acid restriction. And then restricting each of the individual amino acids, branched-chain amino acids, also leads to interesting and significant changes in microbiota composition. Interestingly enough, it appears to not mediate any of the metabolic phenotypes that we've looked at. So we did an experiment where we bladed the gut microbiome using antibiotics, and the late Jay Mitchell's laboratory did an experiment in which they tested the effects of protein restriction in germ-free mice. In both our study and Jay's study, we found that basically all of the metabolic adaptations to protein restriction still happen in the absence of a gut microbiome. There might be, Jay's lab found a little bit of an effect on inflammation, and so that might be worth following up. And all of these studies were done in young animals, so maybe in older animals where the gut intestine might be leakier, there might be more effects of the gut microbiota. But at least so far, we found that even though there are these big microbiota changes, they don't seem to mediate a lot of the effects that we see.
Okay, perfect. Thank you for that. Maybe you could comment on some reasons as to why male and female mice respond differently to, I'm guessing, the low protein diet.
Yeah, that's a great question. So there's a couple, answers that we can sort of think about. So one thing is there might be evolutionary pressures that have set up different ideal diets for both sexes. We see this in Drosophila. It's been shown that essentially male flies have essentially optimal health on a low protein, high carbohydrate diet, while female reproductivity is maximized on a high protein, low carbohydrate diet. And so could there be something similar that's driving this in mammals? Potentially. Another issue is that in our low branch amino acid studies, we found that mTOR kinase signaling was downregulated specifically in males, but not females, by branched amino acid restriction. Now, naively, that doesn't make a lot of sense, right? Because we're restricting branched amino acids by the same amount in both sexes. You would expect both of those to lead to reduced activity of mTOR complex 1, since that's agonized by amino acids. And then mTOR is, a major regulator of metabolism and health, and it's been shown in dozens of studies, if you downregulate mTOR signaling, you get longer lifespan. So we definitely see that there are differences in the effects of a diet on mTOR signaling in males and females. So that sort of just pushes it back one step to ask why. Why does it have these different effects in the different sexes? And that we don't know about. And so there's definitely some things that we're planning to look into this. But unfortunately, a lot of the goadectomy experiments have such massive effects on metabolism by their own that probably there's not going to be a good way to deconvolute that. So we're still trying to think about what the best way to look at that might be.
Awesome. Thank you so much. So metabolic certainly shakes hands with the cardiovascular system. So the next question asks, have you evaluated blood pressure or any other cardiovascular parameter in the mice fed the low protein diet?
So this is an interesting one, and we've done a number of different studies. So we've looked at the effects of dietary protein and branched amino acids in both mice that have a compromised cardiovascular system, so mice that are lacking Lamming or have Hutchinson-Gilford progeria syndrome, as well as in aged wild-type mice. And overall, I can tell you that the effects of dietary protein are very small. That said, there seems to be some interesting issues. There seems to be a trend in general where our protein-restricted or branched amino acid restricted mice seem to have improved diastolic function or improved ejection fraction, but that the heart morphology changed in ways that we might think are unfavorable. But all of these changes are very, very small. And so trying to deconvolute that in the future might be something that is really worth doing. There's a number of labs that are working more closely on branched amino acid levels and the cardiovascular system. So it might be best for some of those to take it up. I will say one interesting paper by Mary Latimer fairly recently at UAB showed that time of ingesting branched amino acids causes or protects from cardiac hypertrophy. So if you eat branched amino acids at a certain time during the day, the mice get cardiac hypertrophy, and at other times they don't. So there might be some additional interesting complicated factors of timing to think about what the effects on the cardiovascular system might be.
Awesome. Thanks so much. A terminology question. Could you explain what IWAT is?
IWAT is inguinal white adipose tissue, and it's essentially a subcutaneous depot that is a good readout of browning and beiging. And so many different stresses, including isoleucine restriction, cause browning or beiging of the inguinal white adipose tissue. None of the mouse adipose depots exactly map to the same depots in humans. But generally speaking, it's thought to be a good model for some of the fat that's in people.
Perfect. Another question for you. Let's sort of zoom it out to the human scale. How do you translate your preclinical finding to humans? Are any of these biomarkers useful?
Yeah, there's a couple interesting things to think about there. One of the questions, of course, is, can you protein restrict or isoleucine restrict humans? And the answer is, of course, you can. One of the things that we have done is look in the USDA food database, and it's certainly true that just as bodybuilders have suspected for a long time that turkey meat is relatively low in branched amino acids, and emu is even lower, but of course less accessible. So people who were eating turkey on Thanksgiving probably were ingesting a relatively low isoleucine meal. So kudos to you. But how is that going to be enough to lead to beneficial metabolic changes? It's unclear. And probably depends how long we're talking about as well. We've done one pilot study, and two groups in Europe have done pilot studies of branched amino acid restriction using medical food that doesn't have branched amino acids in it. There are some limitations to that. It's a powder-based food, so you have to consume it as a drink. It doesn't necessarily taste all that good, no matter what you do to it. But it's certainly feasible to restrict branched amino acids in that manner, and it's been associated in a couple of small studies now with improved insulin sensitivity. Of course, if we find out some of the mechanisms here, maybe we can make drugs that directly engage that mechanism, or drugs that interfere with branched amino acid uptake or catabolism or speed up catabolism. And many people are looking at these as different possibilities. There's also a possibility, one study showed that gut bacteria, depending on how much BRCA transporters they express, they can actually lower blood levels of branched amino acids in people. And so that could be another potential probiotic route for translation. In terms of biomarkers, we could certainly measure blood levels of branched amino acids. They are sensitive to diet. And so I would say that might be, one possibility. But of course, measuring things like HOMA-IR or insulin sensitivity via other mechanisms might be a sort of short-term way of determining if we are effectively restricting branched amino acid levels.
Perfect. That's always a question when we have findings like these: is what are the potential effects, or what are the potential opportunities to translate to humans?
Definitely.
Another potentially final question for you here before we wrap things up. Have you looked at this phenomenon in hibernating animals that have higher brown adipose tissue than mice and rats?
Well, it's an interesting question. No, I mean, certainly there are people here at the University of Wisconsin who work with ground squirrels, so that could be something that we could examine. I'm not sure exactly what their dietary needs are and how we might have to adjust some of our standard diets that work in mice for that. But that's an interesting point. Certainly increasing fat activity in a hibernating animal might, not necessarily be a good thing because they might burn through more fuel more quickly, and maybe not make it through the hibernating period, but maybe that wouldn't happen. And so I think that's a very interesting question.
All right, perfect. Well, I think we'll just leave that as is. Thank you so much again, Dudley, for your presentation and for taking the time to answer all the questions that came up. It was really great to have you here.
Thank you, and thanks to all of the audience members as well for listening, and have a great day.